go ahead and start talking about our new unit. So we're starting Unit 2 of the class. Unit 2 is going to be on histology, the integumentary system, so skin and hair and fingernails and all those slimy secretions. And then we're also going to have the skeletal system. Our next unit exam is the Friday before spring break. My goal is for you to go on spring break and really, enjoy yourselves. I don't want lecture hanging over your head or like the pressure to study for an exam the Monday when you get back. So let's talk about epithelial, just tissues in general. In our bodies, we have lots and lots of different cells. Trillions of cells, 50 trillion cells. And that's a number that's so large most people can't comprehend it. And of those 50 trillion cells, they work together to make tissues, and the tissues of our bodies are going to be grouped into 4 broad categories: epithelial, connective, muscular, and nervous. And we're going to spend the next 3 class periods talking about each of those 4 categories of tissue. Our goal for this class period today is to focus on epithelial tissues specifically. Now, with these tissues, if we take multiple tissues and have them working together for a common purpose, that's an organ. That goes back to the hierarchy of life. And when we look at the study of tissues, the study of tissues is known as histology. Sometimes it's referred to as microscopic anatomy. And there's a fine line, there's kind of a blurred or a gray line that separates histology from cytology, from molecular biology. They kind of all overlap with each other just a little bit. So when we look at these tissues that we have, these tissues in our bodies are made of multiple kinds of cells working together for that common purpose. And these tissues have characteristics that differentiate them from each other. That's how we can tell the difference between an adipose tissue or a compact bone tissue or blood tissue. There are very distinct characteristics, not only in terms of the cells that are present in the tissue, but that stuff outside of the cell, that extracellular material. We call that extracellular material the matrix. And I'm going to camp on this concept for a little bit because I remember it rocked my world when I was an undergraduate. There are parts of tissues that are not alive. I'm going to say that again. Part of our tissues is not alive. Are not alive. We'll say is not alive. So this part of our tissue that is non-living is the matrix. So think back to the last unit, first unit of the class. The cell is the smallest unit of life. If it's smaller than a cell, it can't be alive. But then outside of our cells, we have these proteins, these carbohydrates, these glycoproteins, these macromolecules. They're smaller than the cells. They're outside of the cell. They are non-living. And these are an integral part of the tissues. These make up the extracellular matrix. We have lots of fibrous proteins in our extracellular matrix. Think to lab this week. For those of you who had lab already, think of the areolar connective tissue. It had its elastic proteins, its collagen proteins, lots and lots of big fibers you could look at, or that dense regular or the dense irregular connective tissues you looked at in lab. There's lots of fibers in the extracellular matrix. Other tissue types don't have a lot of fibers. Blood is a tissue that has almost no fiber in its extracellular matrix, but it does have a lot of ground substance. And the ground substance for blood we call plasma because it's super liquidy. So when we think of a tissue, there are 2 key components: the cell and the extracellular matrix. The cell is alive, the extracellular matrix is non-living. They work together to make the tissue. So where do these tissues come from? Well, we started out as a twinkle in our mother's eye and father's eye, and then we were fertilized and we were conceived. And when we were in utero, at one stage, and we'll talk about this more in Bio 314, we were a blastomere, or blastula. And this ball of cells, this big clump of cells, differentiated into 3 layers: the ectoderm, the endoderm, and the Mesoderm. And these three different layers to this clump of cells turned into the different categories of our body, the different kinds of tissues of our body. So when we look at these layers, the ectoderm is on the outside, ecto for outer. Think of the exterior, ecto, outer. And when this outer layer of cells forms the outside of our body. Our skin comes from this germ layer of cells, or the epidermis. And then, if we spend any amount of time studying embryology in Bio 314, you'll notice how our skin folds back on itself to form our central nervous system. And this happens really early on. So we find that the skin and the nervous system both come from the same layer of germ cells, the ectoderm. And then we have the endoderm, that innermost layer of germ cells. And those form the tissues that make up our digestive tracts, our respiratory tracts, our glands. The innermost part of our body is made up of those tissues. And then there's the mesoderm. And the mesoderm was that middle layer of cells that becomes the middle layers of our body, the cartilage, the bone, the blood. This mesoderm we're going to focus on because it forms so many broadly different kinds of tissues. If we think of cartilage versus bone versus blood, for those of you who had lab, the compact, because we covered it this week in lab, the compact bone tissue versus the blood versus the hyaline cartilage, they all have the same source, even though they look dramatically different from each other. One of the challenges that we have with histology is trying to make sense of what we're seeing. We're taking a three-dimensional object and trying to look at it under two dimensions. And I know Ms. Londrich and I have emphasized to you that you should use two eyes on your microscope so you can have that stereoscopic vision. But because of the small size and the two-dimensional nature of our microscope slides, it's still really hard to get a three-dimensional intuition, a 3D intuition about our slides when you're looking at them because they really are in two dimensions. If we take something and chop it up and section it from a three-dimensional object to a two-dimensional slice, that takes some mental gymnastics to translate what you're seeing to what it looked like. Now, we have ways to make this easier. We have fixatives. These fixatives help to keep our slides from decaying because, if we didn't, our slides would smell bad. They wouldn't last for years and years and years. We also are going to use some stains. These stains highlight different parts of our histology slides. For instance, back in January when Ms. Londrich and I were looking at every individual microscope slide that you are working with in lab, we noticed that the skeletal muscle tissues were stained with only one stain. There's pink and then no pink. And it was really hard to see the striations. So we ordered brand new skeletal muscle slides that were trichrome. They had 3 different stains involved. And those 3 different stains really made the striations pop for you. Hopefully you're able to see those striations very clearly during lab. Now as we look at these tissues and we're sectioning them, there's different things that will occur. If we have a centrally located nucleus, sometimes that will be the big dot in the middle. But sometimes I'm thinking of our simple columnar slide, the stomach cardiac slide that you're looking at in lab. Sometimes that columnar cell, even though it's a long tubular cell, when it was sliced from the microscope slide, it was sliced transversely, and it looks like a tiny little circle on the microscope slide, as opposed to that oval that you have in your lab manual, because of the 3-dimensional orientation of the cell as it was sliced to make the microscope slide. This is part of the art form, or the art of histology, being able to look at those slides and figure out, what am I actually looking at? When we think of some of the ductwork, and I love this figure. When we look at common shapes like ducts, depending on where you slice it, it can look really different. So, on the left we have a hard-boiled egg. That hard-boiled egg, if we slice it to the middle, we can see the yolk, or the centrally located nucleus. But depending on how we slice the egg, maybe we won't see any yolk at all, or any nucleus of the cell at all. Or if we look at a macaroni noodle, A macaroni noodle, depending on how we slice it, could be two little circles, it could be kind of a figure eight, or it could be an oval. Trying to take that three-dimensional shape and look at it in 2D becomes a bit of an art form. Next week during lab, we're introducing the integumentary system. And in the integumentary system, we have sweat glands, we have oil glands, we have all these different glandular secretions in our skin. And when we're looking at tissues under the microscope, you'll see a lot of stuff that looks like this. Let's see if I can annotate here. Oh, it worked last week. All right, well, this part of the microscope, or this part of the slide, on the far right-hand corner of the slide, where that tubular duct will look like, almost like spilt milk on your microscope slide. Or, as we look at blood vessels, depending on if we have a longitudinal section versus a cross section versus an oblique section, we can get a very different kind of slice. As you look at the microscope slides that you're working with in lab, you might notice that there's a lowercase cs on the slide. That lowercase cs stands for cross section. I know we have nerve cross-sectional slides, and we also have nerve longitudinal section slides. When we think of the longitudinal sections, those give you a lengthwise slice through that tubular or long organ. The cross-section gives you the shortest possible slice, or a transverse slice. And then anything in between the longitudinal and transverse, we've categorized as oblique. So oblique is kind of the general miscellaneous category. And there are different ways we can make our microscope slides. The blood slides you're looking at are smears. When that slide was physically made, a piece of glass dragged a drop of blood across the microscope slide. It was smeared across the slide with the cover slip. We also could have spreads. And when we make a spread, instead of dragging the material across, it's more like making a peanut butter and jelly sandwich, where you have your microscope slide on the bottom, the cover slip on the top, and you kind of just push it down and wiggle it a little bit, and you spread out the tissues. Our giant multipolar neuron slides are spreads, and our simple squamous epithelial slides that we looked at 2 weeks ago in lab were also spreads. So depending on how the slide is made, we can get different effects. So let's see how we're doing. The histological section that divides a specimen along its long axis is called what kind of a specimen? We have cross-section, oblique, transverse, sagittal, or longitudinal. We're down to about 20 seconds. The correct answer was longitudinal. A slice through the long section of the slide or specimen is a longitudinal slice. And nice, very nice. Well done, guys. So enough about how we make microscope slides. Let's focus on epithelial tissues. So we're going to focus on just epithelial tissues for the rest of this presentation. For those of you who like to follow along in your textbooks, we're in section 5.2 of your textbook right now. So when we think of an epithelial tissue, there's some key characteristics that help to differentiate these epithelial tissues from the other tissue types of our body. For instance, the cells of the epithelial tissues are very closely spaced together. They're like sardines in a can, or like students in Hibberd 100. You're just packed right next into each other. And if these cells of the epithelial tissues are really close together. This means epithelial tissues don't have a lot of extracellular matrix. There's just physically not room for a lot of extracellular matrix in epithelial tissues. Now, there is a little tiny bit of extracellular matrix. By definition, there needs to be to help hold the cells together. But the volume of our epithelial tissues is overwhelmingly going to be cell and the volume of the cells that make it up, as opposed to the volume of the extracellular matrix. Now, when we look at epithelial tissues, you can think of these as the coverings, the linings of our bodies. And there's a surface of it that's going to be exposed to the external environment. We call that upper surface, typically the apical surface, A-P-I-C-A-L, apical surface. And I'm trying not to use that term too much in lab, or at least I was trying on Monday, not to use it too much before we talked about it in lecture. But the upper surface is the apical surface of the cell. So when we're working in lab and you're trying to find the epithelial tissues on your slides, don't look at the middle of your microscope slide. In the middle of that microscope section, you need to look on the edge of the microscope section, because epithelial tissues form membranes or boundaries, and a lot of times they're going to be on the apical surface. Now, when we look at epithelial tissues, most of the glands in our bodies are going to be made of epithelial tissues. We'll talk about glands on Friday. On the note of Friday, if you check the course calendar, it says online for Friday and online for Monday. I'm going to be out of town for a meeting Friday and Monday of this week and next week. So lecture is going to be recorded and posted. Please do not come to Hibbard 100 Friday morning or Monday morning. I am going to be in California. All right. I was waiting to tell you until after the exam. So, back to epithelial tissues. When we look at these epithelial tissues, they're also going to be avascular. Avascular meaning that they do not have blood supply. So if they don't have blood supply, this kind of begs the question, how on earth do they get nutrients to live, to reproduce, to maintain themselves? And the answer to that is through diffusion. Epithelial tissues are usually right next to highly vascularized connective tissues. And that connective tissue it's right next to has a lot of extracellular matrix that has a lot of nutrients diffusing through its extracellular matrix. When we look at the functions of these epithelial tissues, I like to really want to emphasize protection. Our epithelial tissues form the surface, they form the membranes, they form the boundaries, they protect us from the external environment, and they protect us from our own self, our internal environment. We have epithelial tissues in our body that help keep us from digesting ourselves. Or if you've ever spent too much time chewing gum, you might notice how your tongue gets really sore, and that's an example of you wearing off that epithelial tissue that was protecting you. We also use epithelial tissues, if we think of the glandular functions, to release chemical secretions, to excrete wastes. We use epithelial tissues in our digestive tract to absorb nutrients. I'm thinking in particular of the simple columnar cells of the small intestine that have microvilli. We can use epithelial tissues to filter substances, and we also can use them to form sensory stimulations. Now, when we look at these epithelial tissues, particularly the ones that are exposed to the surface, they need to have a very high rate of cellular division. So they're going to be constantly dividing their nuclei, constantly dividing their cytoplasms. And the reason they have such a high rate of cellular division is because we lose millions and millions and millions of these cells every single day. If it was sunny, I'd say go outside after class and try this, but we have to wait for a sunny day. And wait for a sunny day, and it works really well if you can see a beam of sunlight coming in through the window of whatever room you're in, and hold your arm out and just scratch your arm. And you'll see a cloud of white specks float up into the air. And that cloud of white specks are your skin cells. And when we look at dust in the house, most dust in most homes is going to be predominantly dead skin cells that we've been constantly shedding. Yeah, if that doesn't motivate you to dust, I don't know what does. On that note, using a feather duster just throws it up in the air again. You need to wipe with a wet rag. Yeah, I'm not a clean freak. Let's see here. I'm thinking of in our stomach, the lining of our stomach regenerates itself every 3 days. And the reason the lining of our stomach needs to regenerate itself every 3 days is because it's being exposed to a highly acidic environment with lots of digestive enzymes. Our stomach is constantly eating itself. So the epithelial tissues lining our stomach are going to be there to regenerate and protect us. When we look at the epithelial tissues, when we look at the membrane itself, or the layer that's exposed to the surface is called the apical layer, or apical surface, and then the layer that's exposed to the underlying connective tissues is always going to be the basal or basement layer, or bottom layer, as I would say to my high school students. And there's 2 broad categories of epithelial tissues, simple or stratified. When we look at the simple epithelial tissues, it's 1 layer of cells, thick, with a TH at the beginning. So it's 1 layer of cells thick. These simple epithelia are going to have every cell touching that basement membrane. Every cell is going to be involved with the basement layer. And in most of these, every cell will be involved with the apical surface as well. And then we have stratified epithelia. Stratified epithelia have more than 1 layer of cells. There's strata, or layering of the cells. And when we look at these stratified epithelia, most of the cells don't touch the basement membrane, and most of the cells typically don't touch the apical surface. Most of them are going to be somewhere in between, particularly if we have a stratified squamous epithelium. So as we look at these cell arrangements, we can have simple, we could have stratified, look at the blue on the top. But then there's the in-betweeny layer, that pseudo-stratified. Pseudo-stratified looks like there's multiple layers, but there's really only 1 layer. And if we're looking for a technical definition of pseudo-stratified, every cell in a pseudo-stratified layer touches the basement membrane, but not every cell can make it to the apical surface. So we have a bunch of cells that all are connected to that underlying basement membrane, but not all of them extend all the way to the surface. So these cells are of varying lengths. And in lab, we had the pseudostratified columnar epithelium from our trachea that we were working with. We also have shapes that we can have. We can have flattened or squamous. I always think squashed squamous. We could have cuboidal cells, or we could have columnar cells. As a rule of thumb, a cuboidal cell has about the same height and width when you look at it on the microscope. Think of the mammary gland slides that you were looking at. Or when we think of the columnar cells, they are going to be taller than they are wider. The height will typically be at least double the width of the cell. And then when we look at a squamous cell, that squamous cell is going to have very little cytoplasm in it. It's going to have its nucleus, and then it'll taper off and kind of be spindle or fusiform shaped to have almost no cytoplasm left over. So I was getting a little bit ahead of myself, so I guess for the sake of being redundant, I'll go over this slide quickly. The simple squamous is 1 layer of really thin cells. In lab, the simple squamous that you're looking at were the lung sections. We also are going to have simple cuboidal and simple columnar. These are going to be 1 layer of cells each. On microscope slides, cuboidal cells, generally speaking, look round to me. I actually haven't seen a cuboidal slide that had perfect 90-degree angles and a perfect rectangle, or a perfect square to that cuboidal cell. If you see one during lab, call me over. I'll be really excited about it. And then we also have the pseudostratified columnar epithelium. And just to emphasize again, every cell in the pseudostratified columnar epithelium can touch the basement membrane, but not every cell in the pseudostratified columnar makes it to the apical surface, or the top. And a lot of the cells of or tissues of our bodies, particularly in the respiratory or the digestive tracts, we're going to produce a lot of mucus to protect us. And there's a special kind of cell present that makes this mucus. Excuse me. And that's called a goblet cell. It's funny, I was just talking about mucus. And those goblet cells, guys, gals, are supposedly wine-shaped glass, but when we look at them on the microscope slides, I typically think of them as just a clear circle, usually in a simple columnar or a pseudostratified columnar epithelium. So let's talk about each of these individual tissue types. We have simple squamous epithelium, and we need to focus on the apical surface of our tissue slides. If you look in the middle of this slide, we don't have simple squamous. The simple squamous epithelium is that layer of cells along the surface. So on the left, we see the unaltered microscope slide. On the right, we see highlighted the epithelial cells. And this is one of the art forms when you look at the microscope slide. You don't want to focus on the smooth muscle that's present right here. The goal of this slide is to highlight that single layer of flattened cells. And that single layer of flattened cells is going to be a simple squamous epithelium. So when we're looking at our microscope slides for epithelial tissues, make sure you look at the edge of this tissue section. Don't look in the middle. Now when we look at simple squamous epithelium, there's a couple structure-function stories we need to talk about. One of the unifying themes of anatomy and physiology is that structure determines function, or function dictates structure. You know, 6 of one, half dozen of the other. So if we have a layer of thin cells, and it's only one layer thick, as a membrane, it's not a very thick membrane. And it can have very rapid diffusion or transportation of substances. So when in our bodies, whenever we need to absorb something or have that rapid diffusion across a membrane, it's typically going to be a simple squamous epithelial tissue. For our serous membranes, and we talked briefly about serous membranes in the first chapter, these simple squamous epithelials will secrete serous fluid, or the serosive fluid, the liquid that lubricates the organs of our bodies as they move around. We find a lot of simple squamous epithelium in alveoli, and since we haven't covered that unit, alveoli are the little air pockets in our lungs. You should label alveoli air pockets in lungs. So for lab, we have a lung section. Whenever you see a lung microscope slide, I want you to think simple squamous epithelium. We also have simple cuboidal epithelium in our bodies. So simple meaning one layer of cells, cuboidal meaning that the cells are about the same height and width relative to each other. Ms. Londrich, we do have some chairs down here. You can come in and sit down if you want. Oh, okay, all right. Oh. All right, fair enough. We should just not ask any more questions for the rest of lecture. So when we look at these simple cuboidal epitheliums, these are going to be located in many different organs of our bodies. And so it's a cuboidal cell, so there's a lot of cytoplasm present. So we can make some secretions, but it's simple. So it's a relatively thin layer, so it still is going to function for absorption as well. When we look at simple cuboidal epithelium, it's kind of the in-betweeny. It's not quite a simple squamous, it's not quite a simple columnar, so it's going to have properties of both. We can use them for absorption, and we can use them for secretion. Is that my timer going off? Oh, okay, all right. Just want to make sure it wasn't my cell phone. Here's a simple columnar epithelium. This simple columnar epithelium is going to be found in this particular slide from the digestive tract. And I really want to highlight the goblet cells that are present. So here you can see in the unaltered micrograph, large white sections. We have 1, 2, 3 goblet cells present in this tissue slide. And I tried really hard to squint my eyes and make them look like a wine glass, but I just can't. They're kind of like constellations in that they really don't look like what they're supposed to look like most of the time. So when we look at these simple columnar epitheliums, we're going to have one layer of very tall cells. And these simple columnar epitheliums are going to have different functions depending on where they are in the body. This particular simple columnar epithelium from the digestive tract has microvilli along the surface. And these microvilli, which are along the surface, dramatically increase the surface area. They increase the surface area by about 40-fold. And increasing that surface area means that we're able to absorb a lot of nutrients with the simple columnar epithelium. But this absorption of nutrients that occurs with simple columnar epithelium is not going to be where the nutrients go in between the cells. These nutrients are absorbed directly into the cytoplasm of the simple columnar epithelium because of that brush border or microvilli apical surface. We're going to find simple columnar epithelium all over our digestive tract. They are present in the uterus and also in the uterine tubes. I do need to emphasize for you, though, that they won't always have microvilli. This picture just happened to have microvilli on it because it was coming from the digestive tract. Not all simple columnar epithelium will have microvilli. We also have pseudostratified columnar epithelium. When we look at pseudostratified columnar epithelium, particularly the section that's shown from the trachea, is going to have a lot of cilia on it. That cilia functions to move the mucus, which is made by the goblet cells. And particularly in our trachea, we call this the mucociliary escalator. There's this constant stream of mucus being made, shuttled upwards, and then swallowed in our bodies. And the reason we're constantly making this mucus in our trachea is to filter out particulate matter. When we breathe in dust or pollen or mold, we want to get it out of our lungs. Now, because this is a pseudo-stratified or false layered epithelium, every cell touches the basement membrane, but not every cell makes it to the apical surface. We find that we have lots of pseudostratified columnar epithelium in the respiratory tract. And that's the go-to place to find pseudostratified columnar epithelium. Trachea slides have always been my go-to slide for this. There are some portions of the male urethra, which is also pseudostratified columnar. There are also other portions of the male urethra which are actually stratified columnar. But we don't have a lot of male urethra slides. They're pretty hard to come by. So let's pause for a moment, assess our comprehension. Let's say we need to have rapid diffusion of oxygen and carbon dioxide through the air sacs of our lungs, the alveoli. What kind of an epithelium is there going to be? So I've already told you what to associate the lungs with, but let's piece this together. Diffusion means we need to move the chemical across a membrane. And if we want to do it quickly, we probably want a thin membrane as opposed to a thick membrane. What kind of epithelial tissue would function well to quickly move something across its tissue layer or body? We're down to about 15 seconds. Our options are simple squamous, simple cuboidal, simple columnar, keratinized stratified squamous, or non-keratinized stratified squamous. Simple squamous is the correct answer. And nice, solid. Well done, guys, gals. So those were our simple epithelia. Let's move on to our multi-layered epithelia, the stratified epithelia. These stratified epithelia will have 2 or more layers, generally speaking between 2 and 20 layers to them. There is an exception, and that's going to be the thick skin of the hand, the palm, or the bottom of your foot, they can have much more than 20 cells or 20 layers thick. Now, most of the cells in a stratified squamous are not going to touch the basement membrane. Instead, they'll just sit on top of the cell that's underneath, and you just stack layers of cells on top of each other. Because these tissue types have multiple layers of cells that form them, generally speaking, they're going to be thicker tissues or thicker membranes in our bodies. And they're going to have slightly different functions. When we looked at the simple squamous, simple columnar, and simple cuboidal, we had absorption and diffusion of nutrients. When we look at the stratified epithelia, absorption and diffusion is not a common purpose of these tissues because the structure doesn't dictate the function. Instead of being a thin membrane, they form really thick layers. Instead, these form great barriers that prevent the transmission. There are 4 total kinds of stratified squamous, or stratified epithelial. We have stratified squamous, stratified cuboidal, stratified columnar, and transitional epithelium. Now, the stratified squamous is by far the most widespread kind of epithelium in our bodies. Our skin is made of stratified squamous epithelium, and we have a lot of skin. And then if we look at the vaginal walls, if we look at the anus, the rectum, and a lot of the esophagus lining, the pharynx lining, we have lots of stratified squamous epithelial cells. And these stratified squamous are going to have lots of cellular division on the basement membrane. And the reason we have lots of cellular division on that basement membrane is because that's the layer of cells exposed to the most nutrients from that underlying connective tissue. Remember, epithelial tissues don't have blood vessels. And this is a good thing because if we had blood vessels through our epithelial tissues and I scratched my arm, I would start bleeding then because I just ripped off several layers of cells. So this is a good thing, but this means that most of the cells in these stratified squamous epithelial tissues aren't going to necessarily be categorized as live. Their metabolism is pretty much ground to a halt because they don't have nutrients to maintain the chemical reactions of life. Those deepest layers, those basement layers, are the ones that are continually reproducing. As they're reproducing, they push the dead or dying cells farther towards the apical surface, and then finally those dead or dying cells flake off. The technical term for that is desquamation. To desquamate is to remove squamous cells. Most people, though, when you go to the salon, you don't say, oh, I could really use a good desquamation. Instead, you say, I need a facial, I need to exfoliate. They're synonyms with each other. Now, between our stratified squamous epithelial types, the keratinized and non-keratinized, the primary difference is what those flattened cells are filled with. If they're filled with a tough fibrous protein, we call them keratinized. So these keratinized tissues are great at resisting abrasion. Our epidermis on the surface of our bodies is filled with keratin. This is a keratinized stratified squamous. But then if we look at other stratified squamous epithelial tissues in our body, they don't have lots of abrasion. For example, the rectum doesn't have a lot of abrasion, or vaginal walls, or the esophagus. These are parts of our bodies that we do need to have some barriers in, but we don't necessarily have high friction environments. So in those situations, we'll have non-keratinized stratified squamous. So first, let's look at keratinized stratified squamous. This slide is from the bottom of the foot. This should look very similar to what you saw in lab or will see in lab this week. We have stratified in our name here, which means we have multiple layers. They're squamous, which means they are flattened out. And they're keratinized, which means that they're highly waterproof. They're able to resist abrasion. They form an excellent barrier. This would be a terrible membrane to absorb something across. Having multiple layers of waterproof cells would not allow for absorption or diffusion, but it does a great job of keeping stuff out. We also have non-keratinized stratified squamous epithelial. And when we look at these non-keratinized cells, the key thing I want to emphasize for you is how you can see the cell membrane and then the white space for the cytoplasm. If I go back to the keratinized cells, that keratin has filled them all in completely. They kind of mush together. Non-keratinized are much easier to identify on a microscope slide because the keratin doesn't fill up the cell, so you can clearly differentiate cell membrane from cytoplasm and see the outline of the cell much easier. And these non-keratinized stratified squamous epitheliums can still resist abrasion, but they aren't as waterproof. The keratin adds the waterproofing. It's also worth emphasizing that the keratin makes them tougher, so they're better able to resist abrasion, or they're better equipped to resist abrasion when keratin is present. But even if there is no keratin, we have dozens of layers of cells here, and these dozens of layers of cells can still resist abrasion. We have sacrificial layers across the surface right here, and these sacrificial layers of cells can be easily scraped off, sloughed off. They'll serve their purpose by maintaining the integrity of the underlying connective tissues. We also have stratified cuboidal epithelium. These stratified cuboidal epitheliums are going to have multiple layers of cuboidal cells. So when we think cuboidal, think about as long as they are wide. We have lots of stratified cuboidal epitheliums in our skin, particularly in our sudoriferous or our sebaceous glands, or sweat glands that we have. And when we look at these tissue types, we need to focus on the apical surface. Finding apical surface for this slide is pretty easy because there's a big white space on one end. So, this is the apical surface over here. Where's the apical surface here? Finding that apical surface and identifying the epithelial tissue on this kind of a slide is a bit more of a challenge. We need to look for the big white spaces. And the big white spaces in this slide are called lumen, the hollow part of tubular organs. So we have a hollow lumen right here where my mouse pointer is. And then against that hollow lumen, we have epithelial tissues pressed up against the apical surface, or the empty space inside of that tube. We also have transitional epithelium. Transitional epithelium is kind of the in-betweeny weenie, in that sometimes it looks like it's stratified, sometimes it looks like it's simple. It transitions back and forth between having many layers and having almost one layer. And it transitions back and forth as something very specific happens. As we stretch out the tissue, those cells go from being multi-layered to having fewer and fewer layers. And we find lots of transitional epithelium in parts of our body that will regularly distend and then contract again. The classic part, or the classic location in the human body where we typically find transitional epithelium is going to be our urinary bladder, but we also will have transitional epithelium present in the ureter as well. The ureter is the tube that carries urine into the urinary bladder. So when we think of transitional epithelium, I want you to think of stretchy epithelium. To identify this on a microscope slide, the key function or structure I want to emphasize to you is kind of the scalloped appearance of the tissue. It's bumpy or scalloped on the apical surface, and only transitional epithelium is going to have that scalloped appearance. If you look at the other epithelial tissues, they have relatively smooth, non-bumpy surfaces. And that transition from the bumpy to the flattened surface happens as we stretch this tissue out. So, concept check. The blankety-blank epithelium is the most widespread epithelium of the body, whereas the other blank epithelium is rare. So what's the most common kind of epithelium, and what's the rarest epithelium? This rarest epithelium is only present in the male urethra, which is why we did not do it in lab. We actually crossed it out of your lab manuals this week. We don't have any slides for it. All right, let's review the answer. Stratified squamous is the most common. Stratified columnar is the rarest epithelial of the body. And that's all we have for today. We ended just a couple minutes early. I will see you on Wednesday of next week. No lecture Friday, no lecture Monday. Watch the recordings, please.